Hello, this is Bryson. Today I'm going to be talking about the cell cycle, something that you may be familiar with due to mitosis and meiosis, which are parts of the cell cycle. However, I really want to get into detail about specifics of the cell cycle that you probably don't know yet, such as terminology and new phases that you're going to need to know in a higher level biology class such as AP in college. I'm also going to stress the importance of regulation of the cell cycle because without regulation of the cell cycle, life would not be able to survive. So here I have two pictures of cells, which we know are the building blocks of life. Something needs to be cellular in order to be considered living. But what I want to point out with these cells is that they undergo regular cycles of growth and division. And this allows for growth and repair of multicellular organisms, and in unicellular organisms, it allows for growth, but it can also be used as a form of reproduction. This would be called binary fission, and it's when one unicellular organism splits into two cells, which are two separate organisms, and this is seen in bacteria. So here I have a flow chart of what I'm actually going to be discussing in this presentation. So to the left, I'm going to be talking about the phases of the cell cycle that you're going to need to know, which is the G1, the S, the G2, and the M phase. I'm also going to be talking about checkpoints within these phases that allows for the transition of one to the next and the continuation of the cell cycle. I'm also going to be talking about control mechanisms of the cell cycle, which are on the right side. The cell cycle has a complex set of stages that highly regulate it to make sure that it's performing properly and no abnormalities are occurring. And these occur in the form of checkpoints, which are activated by internal and external mechanisms. For internal mechanisms, I'm going to be talking about MPF, which is a maturation promoting factor, and I'm going to be talking about cyclins and these proteins that depend on cyclins in order to actually function, which are called cyclin-dependent kinases, or CDKs for short. For the external mechanisms, I'm going to be talking about PDGF, which is platelet-derived growth factor, which is a protein that is used in the system that humans and many organisms use in order to heal injuries. At the end of the presentation, I'm also going to be talking about what happens if there isn't proper regulation of the cell cycle, and that is cancer. So you're probably familiar with the cell cycle in two phases, interphase and mitosis or meiosis, depending on which cell cycle you're actually looking at. But in an AP or college level, you're going to need to know four phases of the cell cycle. So interphase is separated into the G1, or the first gap phase, the S, or the synthesis phase, and the G2, or the second gap phase. At the G1 phase, the cell is growing and it's developing. At the S phase, it's still growing and developing, but now there's something new that's occurring. It's beginning to replicate its DNA and it's beginning to copy its chromosomes. Once this finishes, the cell enters the G2 phase. The G2 phase, basically the cell is still growing, but it's also finishing the final preparations needed for division. Once all these three phases are completed, the cell can then proceed to the M phase, or the cell division phase. On the arrows, I also have red lines that mark barriers that the cell cycle has. And these are checkpoints that need to be activated in order for the actual cell cycle to continue. So the three marked here are the G1 checkpoint, the G2 checkpoint, and the M checkpoint. Now, checkpoints are very difficult to understand because they're activated by internal and external mechanisms. So the best visual representation I can give is the dial on a washing machine. So when you crank a dial of a washing machine, it begins to turn. And in order to turn, it needs to pass by certain numbers or certain points. And these could be checkpoints. So the G1 checkpoint could be the pump out button or pump out mark. The G2 could be the 40 mark, and the M can be the 30 mark. And these also depend on internal and external stimuli within and outside of the washing machine. If there's no water in the washing machine, a sensor is going to prevent the dial from turning and the washing machine from running because it's not going to allow you to set your house ablaze. And if I don't crank it, which would be an external stimulus, there's going to be no drive for the dial to actually turn. So at the G1 checkpoint, which is considered to be the most important checkpoint in most cells of most organisms because it determines whether or not the cell will replicate the chromosomes and divide, we can see here that it is not active. And since it isn't active, the cell is not going to proceed on with the cell cycle, and it's actually going to go into a phase called the G0 phase, which is the phase in which there is very limited growth and development, if any at all, and it's more of a stable phase in which the cell can actually live and not divide and replicate its DNA, and most cells are in fact in this G0 phase. However, if there are certain signals that activate the G1 checkpoint, then it's going to turn green and the cell cycle is going to be able to continue. The G2 checkpoint occurs right before the M phase, or the division phase. And in order for it to be activated, it needs a protein called MPF, or maturation promoting factor. Well, how do you get this? Well, inside the cell, there are multiple proteins called cyclin dependent kinases, or CDKs, but these are unable to activate the checkpoint. Why? Because they are the improper shape. However, when they bind to cyclins, there is a change in the shape, and this is since the cyclin adds a phosphate group, and this phosphate group actually changes the shape of the actual protein and now gives it a new function, which is able to activate the G2 checkpoint. And this is something called phosphorylation, and it is seen in mostly all of the cellular processes. And when the cyclin and the CDK bind together, this new system is called an MPF, and the MPF is now able to activate the checkpoint, and the cell is able to continue on to dividing and go into the M phase. This is an internal mechanism because the only things that are actually allowing for the activation of the checkpoint are within the cells. There's no external stimulus that is actually calling for the activation of this checkpoint. The M checkpoint occurs within the M phase. It actually occurs right before anaphase because the M checkpoint needs to be activated in order for anaphase to occur. So here we have chromosomes. In the chromosome, there are spots called kinetochores. And kinetochores are where spindle fibers will actually attach to the chromosome to allow for the proper separation in anaphase. When it attaches, it activates a regulatory protein, and this regulatory protein goes through an entire system, which ultimately leads to the initiation of anaphase. Now, if the spindle fiber does not connect to a kinetochore, this regulatory protein is not going to be activated. And what's even surprising is with all of the chromosomes in the human body, if even one is not connected properly, there is going to be no anaphase. The cell cycle will actually just stop right there until it is properly connected. Now, this is, again, an internal mechanism because everything that is activating this checkpoint is occurring within the cell and there's no external stimulus involved. PDGF is an external mechanism, actually, and fibroblasts are used to heal wounds. When you're cut, it begins to proliferate and divide, and this allows for a framework for cell repair, and the wound will heal. But it's not dividing all the time. Why is that? That's because an external mechanism is needed, and this external mechanism is PDGF. So when I get an injury, platelets sense the injury and start releasing PDGF, which then binds to receptors in the cellular membrane of the fibroblasts, and this activates a checkpoint, which then allows for the fibroblast to actually divide, and it eventually leads to the healing and repair of that injury. Now, if there is no regulation, there's something here called cancer, and many people know cancer. They know the term because it's a lot more common now, but they don't understand the definition, and the definition is actually really simple. It is the uncontrolled division of abnormal cells. So usually, if I have a petri dish of cells, like right here, and I wipe away a circle of it, the cells will grow and divide to fill up that gap. But once they fill it up, they're going to stop because they're going to release chemical signals, which is an external mechanism, which states, no, don't divide, we don't have any room left. And then they're going to go into a G0 phase, so they don't divide any further. This is density-dependent inhibition, but cancer cells don't have this. Why? Because there's a transformation. A normal cell turns into a cancer cell when there's some sort of abnormality that prevents the cell cycle from regulating properly. The immune system usually attacks the cancer cell and destroys it, but sometimes it doesn't. And sometimes the cancer cell is able to survive, and then it begins dividing indefinitely. And if you give it enough nutrients, it can divide infinitely. And this will eventually lead to tumors. If a tumor doesn't grow, it is known as benign, which is the better of the two. But if a tumor is malignant, then it is actually halting some sort of cellular process. And this is very, very dangerous and can actually lead to death. And this is when the cancer stages are a lot higher. Now, sometimes cancer can actually break off cancer cells, and they can go to other parts of the body, and these are known as metastatic tumors. And this is a lot more dangerous because now it can be affecting multiple organ systems. So if there's anything that I really want you to take away from this presentation, it's that regulation of the cell cycle is important because without it, we would all have cancer. Our cells would be dividing rapidly, and without regulation, and we would eventually die and there would be no survival of our species. Thank you.